Hi students, welcome to the third video module of molecular basis of inheritance. So basically in the previous two video modules we discussed about the topic DNA and the replication in detail. Okay. So in this video module we shall be discussing the topic transcription. So what is transcription, right? Transcription is basically the production of RNA from DNA. It is called transcription. So basically the process of genetic information from one strand of DNA to RNA is termed as transcription. Okay. So the principle of complementary governs the process of transcription, except the adenosine complements to form a base pair with uracil, because in RNA we know there is no thymine and uracil is present in place of the thymine. Okay. We know replication may, like, we used two strands. Okay, two strands for replication, but in transcription we use only one strand, that is the main template strand for the transcription. Okay, why? There are some reasons. Okay, because firstly, if both the strands act as template, they would code for RNA molecule with different sequences, and in turn they code for proteins, and the sequence of amino acids in the proteins would be different. So एक ही genetic code के बहुत सारे amino acids तैयार होंगे, so that is not feasible. Okay, and the second part is like the one segment of DNA would be coding for two different proteins, right? And simultaneously it would be complementary to each other, so a double stranded RNA will be produced, and this would prevent RNA from being translated into protein, and the exercise of transcription would become futile. Okay, so what are the transcriptional unit? Okay, there are three parts of transcriptional unit, that is one promoter, that is structural gene, and another one is terminator. Okay, so let's discuss about transcription in a great detail. So let's say this is a DNA strand, right? This is a DNA strand, right? We have some portions, right? So let's say this area, okay, this area is known as the promoter. This is known as the promoter, or this is called the promoter. Okay, it's called promoter. Okay, now we have a DNA dependent RNA polymerase, right? So basically DNA dependent RNA polymerase do the same work as a DNA dependent DNA polymerase did in case of replication. Okay, so it has 5 subunits, that is alpha, beta, beta prime, alpha prime, and there is a gamma. Okay, so this is a structure of a RNA polymerase. Okay, and with this there is a sigma factor. Okay, so sigma factor joins with the RNA polymerase on the promoter side, then the transcription starts. Okay, the transcription continues and the complementary RNA starts getting produced, A, T, T, A, G. So here we will produce U, A, U, C, like this. Okay, so after the so when the process needs to be terminated, a thing which is called the row factor, that is also called termination factor, that joins here and the single strand of RNA is produced. Okay, remember the whole process is similar to the replication, because the process continues from the 5' direction to the 3' direction. Okay, it is 5' and this is 3', and the template strand is 3' to 5'. Okay, and the fun fact is the strand which is not coding for anything is known as the coding strand. Why coding strand? Because the coding strand ka sequence will be complementary to the template strand ka sequence, right? It will be complementary, and again the RNA is produced will be complementary of this template strand. So basically the new RNA and the coding strand have the same complementary RNA pairs. So this is called the coding strand. Okay, you can see here DNA dependent RNA polymerase catalyze the polymerization in only one direction, that is 5' to 3' direction, and the strand that has the polarity 3' to 5' act as a template strand and also referred to as template strand, right? Strangely, the strand which doesn't code for anything is referred to as coding strand, because all the reference point while defining a transcription unit is made with the coding strand, right? To explain the point, a hypothetical sequence from a transcription unit is represented below. As I said earlier, कि इसका complementary ये होगा, and the new RNA produced will also be complementary to the template strand. So the both complementary strands, their DNA, their base pair sequencing will be similar to each other. Okay. The promoter and the terminator flank the structural gene, right? To the 3' end there is presence of the promoter, and to the 5' end there is presence of the terminator, because right. The DNA sequence coding tRNA or RNA molecule also define a gene, however defining a cistron or segment of DNA coding for a polypeptide. Okay, so basically two things, one is monocistronic and another is polycistronic, right? So the coding sequences or express sequences are called as exons, and exons are said to be sequences that appear in mature processed RNA. Okay? The exons are interrupted by the introns. Okay. The split gene arrangement further complicates the definition of a gene in terms of a DNA segment. So what are the types of RNA? There are three types of RNA. One is mRNA, another one is tRNA, another one is rRNA. Okay. So basically three types of RNA are present here, right? So there is a single DNA dependent RNA polymerase that catalyzes the transcription all types of RNA in bacteria. That means the opening or unwinding, all works are done by the DNA dependent RNA polymerase. Okay. As I said, key initiation factor will be sigma and the termination factor will be rho. Okay. But it is in the case of the prokaryotes, right? But in the case of the eukaryotes, what happens? Key there is a clear division of labor. RNA polymerase 1 transcribes rRNA like 28S, 18S and 5.8S. This is very important question. Very important question that has been many times asked in the NEET exams. The second is the RNA polymerase 3 that responsible for the transcription of the tRNA, 5' RNA and snRNA. Okay, just remember the names. And the RNA polymerase 2 transcribes for the precursor of mRNA, the heterogeneous nuclear RNA or the hnRNA. Okay. The second complexity is that the primary transcript contain both the exons and the introns, so there is splicing. Basically splicing में क्या होता है कि introns को हटा दिया जाता है and exons को join कर दिया जाता है. So जब transcription हो तो वहाँ बीच में कोई intron ना आए, right? And there is also process called capping. So capping कहाँ पे होता है? Like to the 5' end of the hnRNA, that is done by the methyl guanosine triphosphate. MGT, methyl guanosine triphosphate. This has been asked sometimes. So basically this was all about your transcription, and you need not to worry and do some MCQs, right? A transcription unit of DNA is defined primarily by the three regions in DNA. Which of the upstream and downstream these are? तो एकदम डाउनस्ट्रीम में क्या रहता है? एकदम डाउनस्ट्रीम में रहता है तुम्हारा प्रोमोटर, देन स्ट्रक्चरल जीन, देन टर्मिनेटर, राइट? प्रोमोटर, स्ट्रक्चरल जीन, देन टर्मिनेटर. ओके, promoter, structural gene, then terminator. So okay, this was all for the video transcription, and I have make you understand the whole transcription process just in some 10 minutes. Okay, so do like, share, subscribe the video, and stay tuned with us. We will finish the syllabus within the span of 1 month before the NEET 2025. Okay, thank you.